So it's my first time in Israel. My impressions are the weather is amazing. The food is even more amazing than the weather. And I like the relaxed feel of the people here compared to New York, which is very kind of angry and frenetic. So I don't know if this extends in all directions from Jerusalem, but it's great. All right, as Kaya said, I spent the first 20 years of my career working on Drosophila, which has a lot of genetic tools and a lot of beautiful behaviors, beautiful neural development. But in 2008, the big midlife crisis hit, and I thought, what am I going to do for the remaining decades of my career? And so I thought that the study of how behavior emerges from brain circuits is best done in an animal where you understand what the business model of the animal is. And so if you look at this animal, so this is the female of the mosquito Aedes aegypti. Her business model is very clear. For her to reproduce, she needs to find one of us in the audience, land on our skin, break the surface of the skin, push around to break capillaries, and then drink a huge blood meal. So if she's unable to do this, she won't reproduce. So that for her is a very good behavior. That must emerge somewhere in the circuit. She has to sense us. She has to be able to fly toward us. And it's a very bad thing for us. Because in doing so, if the person before who was bitten was infected, she'll infect you. So I'll tell you two unpublished stories. So first, some background about the biology of this animal, how they integrate multimodal cues to actually show these behaviors, and then very, very fresh new data, the first attempt to look in the mind of the mosquito to see how these cues are actually activating brain circuits, and then at the end, a developing story about how they sense temperature. So here's the behavior in action. Here's a couple centimeters of my student Emily Dennis's skin, put into a cage of female mosquitoes who have never had a blood meal. They immediately figure out that there's a little patch of skin that is giving off body odor and heat. The skin is covered with delicious substances, and you can see the movie sped up a little bit. They right away land on the skin, puncture it, and they're filling up with blood. So they'll triple their body weight over the course of a 10-minute meal. And the results of this, of course, are familiar to you, that it's painful, it's itchy, and so again, really bad for humans, really great for mosquitoes. So unlike Drosophila, these guys go through a terrestrial cycle when they're flying around biting people, and an aquatic cycle when they're developing, which is why you should eliminate standing water around your homes, because you're only helping them breed. So there's lots of fascinating behaviors at various points here. So the males and females mate on the wing, so in the air. It's an amazing behavior. They have to find each other and then mate in the air. The female then develops this really intense drive to find humans, this intense hunger. The intensity of the behavior is what drew me to study these. Like they're very, very focused on doing this. And as soon as they have a blood meal for the next 3 days, they completely lose interest in humans. So it's a behavior that you can turn on and off with a blood meal, and she'll do this repeatedly throughout her life. At the point that the eggs are ready to be laid, she develops an extremely strong attraction to water. So you switch from being attracted to humans to being attracted to water. The eggs are laid, and then they develop over the course of about 10 days, and then the cycle repeats. And it's like a transaction. One human blood meal, 2 or 3 microliters of your blood produces 100 mosquito eggs. So every time you're bitten, you're helping to populate the world with new mosquitoes. And they're very successful at doing this. And so she'll be able to bite multiple people in the course of a lifetime, of course, spreading disease with each bite. So not only is the sensory neurobiology really cool, but it's also really important for human health. So this is a map of a couple months ago of the incidence of diseases spread by Aedes aegypti. So yellow fever is an old disease for which we have a vaccine. Dengue fever, we don't have a vaccine. Chikungunya, which was unheard of in North and South America until a year and a half ago, are spread by these animals. So just 2 examples of how good these guys are at finding humans and spreading diseases. So the first time chikungunya, which comes from East Africa, was ever found in the Americas, a plane landed in St. Martin, either an infected human or mosquito got off. So the first case was in December 2013. By September 2014, so just 9 months later, there were 1 million cases in the Americas. So from 1 case to 1 million within a calendar year. And here's a virus that came from this part of the world. So West Nile was first found in the US in 1999. Again, airport related, so around JFK, either a person or a mosquito got off the plane, infected people in Metro New York. Next year, it started spreading, the next year, and then within 3 years, it's spread across the US. So mosquitoes are really, they love the jet set life of humans, and they're really good at following humans around the world and spreading disease in their wake. So what's really important about their capacity to spread disease is that they are extremely selective in biting humans. So this is a field study by Laura Harrington from Cornell, where she sampled mosquitoes that are living in and around the village and asking what kind of blood have they ingested. So you collect mosquitoes that have taken a blood meal, you squish them, and then you use immunotyping to figure out what kind of animal they've bitten. And although the village had a large census of domesticated animals, the mosquitoes essentially ignored the domesticated animals and went very selectively for humans. So I'm a sensory neurobiologist trying to understand the link between genes and behaviors and the circuits in between. So we're studying all aspects of these behaviors, so how we look, the smells we give off, so humans are very smelly. CO2 that we exhale is also a potent attractant for mosquitoes, and when we're alive, we produce body heat. So the mosquitoes pay attention to all of these cues. And you kind of don't have a choice in releasing most of these, so if you're alive, you'll be emitting heat and CO2 and odor. And so here's an example of how human odor differs from animal odor. So if you collect, if you make a human wear a nylon stocking, or you make a guinea pig wear a nylon stocking, and then you do chemical analysis of the odor that has soaked into that nylon, you get different chemical peaks, and you can see that for an odor like this, the guinea pig makes a lot of this molecule and humans make very little, and then vice versa. So this is a kind of a fingerprint of smell, like a perfume that animals give off that differs from the smell that humans give off, that mosquitoes that prefer humans can figure out. So it's a great neural transformation of, this is just a part of the hundreds of odors we give off, so the mosquitoes know that it's us and not animals. So, so how do they know that we're human? So there must be this interesting transformation. They're after the blood, so they're trying to figure out what kind of blood are they going to be getting. And the reason that we have this great body odor that mosquitoes love is because of the skin microbiome. So the skin is coated with a garden of different bacteria that feed on the sebum and other fatty acids that humans give off, and that gives us uniquely human body odor. So it isn't just that the mosquitoes are going after humans over non-human animals. There's also a lot of evidence, both scientific and anecdotal, that not all humans are equally attractive. So you may have experienced this if you're outdoors in a social grouping, that one person will be particularly persecuted by mosquitoes. So we've done this in our lab in New York, and it's absolutely true. But there's also been field experiments in East Africa where you have people sleep in different rotating huts overnight, and then you come in the morning and figure out how many mosquitoes were attracted to the person. You repeat it multiple days in different huts. And the results here are very striking, and we found exactly the same thing in laboratory experiments, that if you look at different subjects over the course of many different days at different locations, a person like subject A attracts an average of about 50 mosquitoes a night, and person K attracts about fewer than 10. So there are these huge interindividual differences that there's a huge folklore around this. If you type this in, there's all sorts of ideas that are either have some credence or are ridiculous. But and so we and others are actively investigating this. So there's something really interesting about the ability of mosquitoes to select one person over another. And so there is something important here. If we could figure out why they're targeting one person over the other, we have a kind of a cue about what is it that makes us attractive. So in the first part of the talk, I'll go over the amazing integration of different sensory cues that mosquitoes use to find us, focusing on carbon dioxide as a cue. So what happens if we make a mosquito where it's no longer able to detect carbon dioxide, where it can still only respond to our body odor, heat, how we look, and moisture coming off of us? So first I have to tell you about how mosquitoes sense carbon dioxide. So there are 3 different membrane proteins that we have no idea exactly how they sense carbon dioxide. I was really excited last week that after centuries of work, someone has finally figured out how we sense carbon dioxide in blood when we're hypoxic. So that's finally been decoded for humans. And humans use a totally different pathway. But so insects use this combination of 3 different membrane receptors that somehow convert CO2 detection into spikes in sensory neurons. So there's 3 subunits. We've worked on this in Drosophila, so we were pretty sure that this is an essential subunit to make a functional CO2 receptor. And so we wanted to start doing genetics to try to get at what's the link between genes and behavior. So of course there's a really long history, century of history of doing this in this creature, Drosophila melanogaster. So the first transgenic mosquito was made, the first transgenic fly was made 30 years ago, and the genome was sequenced in 1999 in Drosophila, so flies have a huge head start. I decided, like in the mid-2000s, that the mosquito was almost ready for the transition to do real genetics. And so you can see the first transgenic mosquito was made many years after the first transgenic fly. RNA interference is possible in mosquitoes. So the feeling was you need to have a genome to do genetics. It's extremely helpful. And you need to do genetics, meaning go in and knock genes in and knock genes out. And so this is the point that my lab entered the field in 2008, and we and others began making knockouts 5 years later. So in the beginning, we used zinc finger nucleases, now a deeply uncool method to make mutants. Of course, everyone is using CRISPR-Cas9, and so are we. But in this earlier work, we used the much more expensive protein-based mechanism. So you design a protein that will bind to either side of a gene that you're interested in targeting, and you can either make a small deletion, or you can integrate something into the locus to tag it. And so my postdoc, Connor McMenamin, knocked out the gene on the left, GR3, leaving the 2 other subunits intact, but presumably non-functional. And this was published last year in Cell. So what's great is that he cut the chromosome at the site of this gene and then put in a ubiquitous marker. So the mutants are brightly fluorescent with ECFP, which you can see here, a pupa that's blazing blue with fluorescence. Okay, so what's wrong with these animals? So what's not wrong with them is that they aren't debilitated in some way with locomotor difficulties. So we fly them in this box, and we can track how good they are at flying and whether they abide by the rules of a diurnal animal. And so you can see here's the flying behavior of these animals over the course of 24 hours. The wild types have flying activity that ceases when the lights go off, so they're presumably sleeping here. And then at their dawn, they wake up again. And so the mutants, where we've knocked out both copies of this gene, are normal. They have normal activity rest cycles and normal flying. And now we look at activation by carbon dioxide. So mosquitoes love carbon dioxide. The worst thing you can do is breathe, because CO2, you're exhaling 4% CO2. So we have this amazing 3D tracking system where you can release females or males into this box. We have cameras that can track in real time their flying behavior. So this spaghetti here is 6 minutes of females just flying around with nothing in the box. Then we inject a little bit of CO2 and they go berserk. So there's a huge increase. This is time. This is the amount of distance where they're flying. So CO2 causes this immediate activation of the animals. If you eliminate 1 copy of this gene, they're normal. There's no effect. But very strikingly, you eliminate this 1 gene in the genome of 14,000 genes, and they're completely unable to sense carbon dioxide. So they don't really care. There's no difference in flying behavior. So we have made a CO2 deficient sensing mosquito. So now we get into multimodal integration. I told you that these guys care about heat and CO2 and odor. And so this is how CO2 interfaces with these other cues. So here's a video of a bunch of mosquitoes hanging around doing nothing at ambient temperatures. We now heat a little Peltier here, inject CO2 into the chamber. Two things are happening. They're going berserk and flying around a lot more. And they sense that this invisible, there's this invisible target here, but that's warm to 37 degrees. So they're really, really good at seeking out this spot that's warmer than the other walls of the chamber. And what they're doing when they get here is they have this illusion in their heads that there's a human arm next to it. So they're putting their proboscis through the mesh, trying to find a person to bite, even though there's nothing on the other side of the mesh except a metal Peltier giving off heat. So just these two physical cues are enough to trigger this really innate behavior. And my student Ramon Corfas has found a way to automate this, so to be able to score the number of mosquitoes that are landing on this warm Peltier. And these behaviors are critically dependent on the provision of both CO2 and heat. So if you have only a heated Peltier, they don't go. And if you have only CO2, they don't go. So it's these two cues together trigger the behavior. And very pleasingly, the mutants don't do anything. So they don't, even though they're flying normally, they have no idea that there's heat there. So even though they're perfectly capable of sensing heat, the inability to sense CO2 makes it impossible for them to sense heat. So how do these mutants respond to behavior? This is my former postdoc, Lindy McBride, showing how the two-portal effectometer works. So you have a choice assay of something in the first port, something in the second port, and then we quantify how many animals fly into this or that trap. In this case, it's Lindy versus a guinea pig. So in these experiments here, we look not at Lindy or guinea pig, but a very abstract representation of a human, which is the small molecule lactic acid. So it's a fatty acid that we give off when we sweat. So it's a characteristic odor of humans who are sweating. And so what's really cool is if you put lactic acid into this trap and nothing in this trap, if you don't supply CO2, and you look at the number of animals that are responding to the stimulus side or to the blank side, nobody cares about anything. Now you put in lactic acid plus CO2, and you can see that the wild type guys, they love the lactic acid. They ignore the blank port, as do the ones that have one functional copy, and the mutants don't sense the lactic acid. So it's, again, unless you're able to sense CO2, you have no idea that there is this delicious human odor there. So multimodal integration of carbon dioxide with heat, multimodal integration of carbon dioxide with lactic acid. And you see the same thing with a much more complex stimulus, which is if you guys were to take those of you wearing socks, if you were wearing socks, socks have lactic acid plus hundreds of other odors. So again, if we put a sock in here, there's this huge, socks alone are sort of mildly interesting to everybody, but only the ones that have a functioning CO2 pathway are massively stimulated to go toward the dirty sock, and the mutants, again, are impaired. So being able to sense CO2 is really key to the hunting behavior of mosquitoes. So I'll show you a couple more behaviors, so getting progressively more realistic to what the mosquito's doing when she's flying outside. So again, here, the behavior of these guys is to find a blood meal. So we can do this artificially by taking a big vat of blood, stretching a parafilm membrane, and if you heat up the blood, the females, and you supply CO2, the females will go, and a high percentage of them will do this, which is they'll puncture the membrane as if it's skin and drink. So if you supply both warm blood and CO2, you get a lot of feeding. Warm blood alone, they ignore it. Room temperature blood and CO2, they ignore it. So this is a real, again, fusion for them it's a really smart thing to find a live human that has blood that's flowing and not coagulating. So how do the mutants fare in this assay? So here we have warm blood and mosquitoes activated by CO2, and the wild type and the one lacking one copy of GO3 are very good at blood feeding, and the mutant is unable to do so. So again, you need CO2 to do this behavior. But I love this experiment because you can take an animal that's crippled and unable to sense CO2 and just provide around the edges another stimulus, which is the dirty sock. So you wrap a dirty sock around the artificial blood feeder, and now you can restore the behavior. So now a fusion of warm blood and body odor, you can get these animals paying attention and blood feeding. So how does this work in a real-world situation? So my postdoc flew halfway around the world to fly the mutant mosquitoes in these field cages, which are in a tropical region in northeastern Australia, where there's a lot of native Aedes aegypti mosquitoes and then periodic outbreaks of dengue fever. So the way these experiments look is that we take advantage of the fact that the mosquitoes who are mutant are fluorescent, and so we can fly a mixture of wild type and mutant mosquitoes into this enclosure and see how good they are at finding humans, like a treasure hunt of there's a human sitting either here in this sort of house structure or sitting out here in the yard, and we can ask how good they are at finding humans. So the human sitting in this cage, it's really warm. Human is sweating and breathing and has visual cues coming off of them and is smelly. So is the lack of ability to sense CO2 important for these creatures? Here's our high-tech behavior assay. So we have a person with naked legs sitting, in this case, sitting in the house. There's some dirty laundry here. He's breathing. This is Connor. And every moment that there's 200 mosquitoes in this cage, 100 are wild type and can smell CO2, and 100 are mutant. So he aspirates one of them as it lands, and then we genotype them afterwards to figure out who has found Connor. And they do amazingly well with this. I found this result both amazing and really depressing, considering how much money it costs to fly all this stuff around the world. So in blue is the behavior across time of the mutants. In black is the wild types. So cumulatively, he's capturing as many of the mutants as wild types in these experiments. If you put him a little bit further out, away from the house, fewer mosquitoes are out there because they like hanging around houses. That's their natural behavior. And you see a slight but significant decrease in the ability of the mutants to find Connor at this distance. But still, they do amazingly well. Again, hammering home this point that they are not idiots. They don't rely on just one cue to find humans. You take away one sense, in a very naturalistic environment, they will rely on other senses. So in this last experiment, we modeled this biology by using mice. So mice are not the preferred host, but given no other options, the mosquitoes will be attracted to them and will blood feed on them. So we do the same experiments that we did in Australia, which is release both wild type and mutant animals, give them a mouse, and then ask how many find the mouse and blood feed. And so we did this. The idea would be that you make it harder for the animals by putting the mouse either in a small cage where it's very obvious that there's a mouse sitting on the bottom, versus a very large box, sort of the equivalent of 2 or 3 soccer fields with a single mouse on the bottom. And so if you have a small cage, the mosquitoes, both the wild type and the mutants, are equally able to find, so 50% of the mosquitoes find the mouse and blood feed on it, and there's no effect of the mutation. Now you put the mouse into this huge enclosure, and you see this very dramatic effect. So at different length scales, it starts to matter whether the animals are deprived of the ability to sense carbon dioxide. So what I've shown here with this series of experiments is that CO2 is critically important for these animals to sense odor, as well as heat. And experiments I didn't show moisture. So this is the major integrator of behavior that allows these animals to behave. So the question that I've been trying to ask since we published the paper is, how does the brain do it? Where in the brain how is CO2 sensitizing the brain of the mosquito to do this? And so I'm really excited. Like in the last 2 weeks, we started getting some traction on this. So to look at the circuits, you have to go into the mosquito brain. Mosquitoes, just like primates or humans, have different brain structures with different functions. So here's a cutaway of the mosquito brain. You have visual areas, you have lower level olfactory and gustatory areas, and then up here is the sort of mosquito equivalent of hippocampus and cortex, to the extent that these guys have such areas. So we know exactly where to look for where heat would be received, where body odor would be received, where CO2 would be received. And so in the initial experiments, we are focusing on the antennal lobe, which is where odor cues come into the system. So you have, this is the equivalent of the nose in insects. So we have the antenna, the maxillary palps. So odor information comes in through the antenna, carbon dioxide comes in through the maxillary palp, and then we can look, do we see where in the brain are these cues activating circuits? And so my postdoc, Meg Younger, who's been in the lab for a year, her task was, all right, get a prep where we can do calcium imaging in these insects, and make a transgenic mosquito where we can get Gcamp into the mosquito so we can monitor neuronal activity. And so her prep involves cutting a little window in the cuticle of this animal, and then just taking a look to see activity. So this is took a couple of months to get working. The trick is that you want to expose the brain. So this is the brain bathed in insect saline, so we can have a look down. But we want to keep the sensory structures dry so we can start puffing on different stimuli. And the mosquito's really happy. It will sit there for 4 or 5 hours while the activity of Gcamp can be monitored. So the next task was for Meg to make a mosquito that expresses Gcamp. We saw earlier the raw genetic power of the mouse, where you can go in and make neuron-specific knockouts. It's incredible. So mosquito, we're not there yet, so we're starting with the good enough experiment of just putting Gcamp into every cell. Obviously, it's like early days. The more refined experiments of putting it into interneurons, inhibitory interneurons, or projection neurons or olfactory sensory neurons remains to be done. But the initial glimpse, this is the antennal lobe of these animals. So we want to figure out, can we detect odor-evoked activity in these regions? And so here again, the experiment is that you puff on lactic acid, the odor that I showed you that evokes very little behavior unless CO2 is present. And so here's kind of a hard to see movie because it's very bright on the screen, but there is a glomerulus that is lighting up when lactic acid is puffed. So this is the whole structure of the antennal lobe. Here's some nonspecific fluorescence here. This is the most exciting thing I've seen in the last year. Okay, there's other exciting things I've seen, but it really is an amazing achievement. I'm really happy that Meg pulled it off, because no one has ever done this before, and I think you have to get in and look at neural circuits if you want to understand how the brain works. So here's 3 different trials of puffing on lactic acid. So again, you get highly selective activation of this and no other regions of the brain. So if you look at this region of interest and quantify fluorescence, you can see that before lactic acid has been puffed, you have a baseline, and then there's a huge increase when this molecule is puffed on, and then it goes back down to baseline. A control odor, benzaldehyde, which is a plant odor, elicits little or no activation. And then even after this talk was done, Meg found a glomerulus that's activated by benzaldehyde, so, which is in a different region from that of lactic acid. So we're fully 15 years behind where we are in Drosophila. We hope to catch up in the next 3 months, right? So the goal is to make a complete map. So all of these structures are points of convergence where olfactory neurons tuned to different odorants, be they plant odors or odors, human odors, come together into these different relay centers, which are then sent up to higher brain for the animal to figure out, is it a plant, is it a human, is it a guinea pig, or is it particular humans? And so the predictions based on our published work is that CO2 should activate, say, this hypothetical glomerulus, lactic acid, this now known glomerulus. So we should see some mechanism of synergy somehow. So whether it's presynaptic facilitation there, some effect on the inhibitory interneurons that release a block on behavior, something. And then the holy grail will be to pump on human odor and see how you go from these sensory inputs to these behaviors. So I'm really excited about that. We will catch up really quickly, so pretty juiced about it. The other thing that's cool is the more, we started in 2008 and people thought we were insane. Nobody was working on the mosquito using neurogenetics, and just in the last month I found 3 other labs who are also making Gcamp mosquitoes. So it's a field that will go from the most trivial fringe investigation to something that will be a little bit more central, so it's exciting. So in the last story I'll tell you our investigation about heat sensing. So I understand it gets really hot here at some point in the year. Nevertheless, the mosquitoes have to hunt you guys. They have to be able to find you out in the wild. So I already showed you these data just to remind you that, CO2 activates flying behavior. And just to review that in the presence of CO2, you get this heat seeking. So here's the behavior of a female going on to that Peltier I showed you. And if we have the Peltier off, the female doesn't go. If we get rid of CO2, the female doesn't go. And I didn't show you this before, but males don't go. So here's the case. Males have a totally different business plan. Males eat from flowers and they mate with females, and that's it. That's all they do. And so they don't care about human heat because they don't bite humans. So there's a great deal of dimorphism in behavior. So let's look a little bit more at these responses to heat in the presence of CO2. So in each of these cases, this is Roman Korfas, has taken a bunch of female mosquitoes and he's just kind of playing with them. He heats up the Peltier to 40 and then cools it, heats it, cools it, heats it, cools it, and then looks at the percent of mosquitoes that go to the Peltier. So they go, they leave, they go, they leave. He just torments them for hours and they never get sick of this game. So they're really, really focused. There's no adaptation in the behavior, as there shouldn't be, because if they don't find blood, they won't reproduce. And so you can see over the course of these 12 stimuli covering more than 2 hours, there's no variability in the behavior. So now as he ramps up the Peltier from ambient, which is 26 degrees, to very hot, 60 degrees Celsius, these are representations of the side of that cage where we're looking at quantifying the number of mosquitoes on the Peltier. So these are heat maps of the occupancy of mosquitoes over the course of the experiment. You can see when the Peltier is set to ambient, they're not interested. As you heat up the Peltier, the maximum occupancy is at 40 degrees, because that's us. They really like us. They really like hanging out at 40 degrees. Once you heat the Peltier to these temperatures, again, they're not idiots. 55 degrees is very, very warm, and so they leave. And so that is mapped here. So these are, again, playing around with ramping up the temperature of the Peltier here, and then looking at the attraction of mosquitoes. Here's our control. They love 40. They ignore 26. As you increase the Peltier, you see a peak here around 40 degrees, and then they leave. So this is the tuning of these animals. No other insect does this because it's a really specific behavior for animals hunting humans, to go for human body temperature. So in a series of really clever experiments, Roman asked, what is it that they're paying attention to, the absolute temperature or the relative temperature? So if he cools these animals, cools the assay down to really cold, 21 degrees, will they still wait for 40 degrees before they go after the heat source, or are they paying attention to the relative above ambient? And so the answer is it's the relative above ambient. So in a room that's warmed to 21 degrees, even at 2.5 degrees above ambient, so this is 23.5 degrees Celsius, they're heat seeking, even though objectively it's not heat. It's just they're able to integrate what's the ambient temperature and what's warmer relative to ambient. So if the room is very hot, they still heat seek 2.5 above ambient. And so that's mapped here. So you can get heat seeking here at these temperatures very, very low relative to what they would normally do. So if you look at the delta temperature above ambient, you can tell that they really are calculating these differences above the room that they're sitting in. So depending on the room temperature, do they have increased tolerance for hot temperatures? And the answer is no. So when you heat the Peltier very hot, regardless of the temperature of the room, they know to avoid a 60-degree Peltier. And so that's shown here, is that there's an absolute offset at which these animals are avoiding hot temperatures. So we think that there's two distinct mechanisms that mosquitoes use. They need to focus their behavior into about 40 degrees. They need to be interested as the temperature increases above ambient, but they need to know when it's too hot. So there's some absolute threshold for avoidance, and then some progressive delta T interest in going to humans. And so we started looking at the genetics of this. So this is a snake plot of the TRPA1 ion channel. So this is a non-selective cation channel that's permeable to calcium, that has all of these anchorin repeats that respond to chemical agonists. And this is also a thermosensitive ion channel. So if you heat up membranes, it will start fluxing calcium. And so Raman made a mutation that eliminates the function of this gene. And so again, he can tag it, tag the mutants with ECFP, so we have null mutants. And we can show, I mentioned that there are these anchorin repeats that are activated by chemical agonists. So N-methylamide is one such molecule that irreversibly activates the ion channel, and animals don't like drinking it. So if we do drinking experiments where we give them a choice of sucrose or sucrose poisoned with N-methylamide, the wild types don't like the taste of N-methylamide. Even at 10 millimolar, they avoid it. Whereas the mutants have no idea that the water is poisoned with N-methylamide and with sucrose, and so they drink equally from both the poison sucrose and the regular sucrose. So we think there really are genuinely null mutants. So how do these guys interact with heat? Does this gene play a role in this part of the behavior going toward warm, or this part going away from heat? And so the answer is it's that they need this gene to be able to avoid very high temperatures. And so that's shown here, that the heat seeking is normal in the mutants in purple, but the mutants are unable to avoid these high temperatures. So they're still really interested in a very warm Peltier. And then that's summarized here. So we think that TRPA1 is critically important for these animals to avoid high temperatures. And in the last experiment, using a gradient, we showed that that's absolutely true. So if you put these animals in a gradient that goes from cold to warm, and we like to see where the animals like to hang out, from cold to hot, you can show in this time series when the Peltier is quite cool, with it progressively warms up to higher temperatures here, and we can put in a wild type, a heterozygote, or a mutant, and you can see that the wild types immediately figure out that this part is cooler, and they congregate here. The mutant is much less able to make that calculation, and many of them end up here in the very warm part of the Peltier. So if you look at the evolution of this behavior over time, the wild types avoid the hot side, go toward the cool side, and the mutants really has a hard time figuring out where to go. As a result, you find a lot of them accumulating here at the very warm side of the Peltier. And here's my deathogram. So if you look at what those animals are doing at the right, the mutants are dying in droves when they end up there. So the inability to avoid high temperatures, to sense it, means they accumulate on this hot area and then they die. So this is a picture, a movie of New York on a hot summer day, so people walking around, photographed by Raman with a thermal camera, and you can see here there's regions in the environment that are very warm. These people are giving off different degrees of heat. So the mosquito somehow has to figure out where is the appropriate host temperature, has to be attracted toward areas like this, but to be able to avoid areas in the environment that are very hot. And so we think that TRPA1 is a key molecule that helps them make that calculation. So the work I told you about today on CO2 sensing was led by Connor McMenamin, who just took a job at Hopkins, supported by Roman Korfas and Ben Matthews. The heat-seeking work unpublished by Roman Korfas with these folks, and then Meg Younger, who with a Grass Foundation Fellowship last summer, started the process of being able to look into the mind of the mosquito. So thank you very much.